You know, right now, at this very moment, there's a war going on inside your body. It's a massive, invisible battle against millions of invaders, and honestly, your life depends on the outcome, every single second. So let's pull back the curtain and take a look at this incredible internal defense system.
So the battlefield is your own body, but you can't see any of it. It's all happening on a microscopic level. So how in the world do we find the clues? How do we know if our internal army is actually winning the war, or if an enemy has slipped past the defenses? Well, to figure that out, we have to become detectives.
Every great army has different branches, right? And yours is no exception. First up, you've got Division 1, the Sentinels. This is your innate immunity. Think of them as the guards on the wall. Your skin, certain cells, they're always on patrol, ready for an immediate response. They don't care who the enemy is, they just attack. But then, you've got Division 2, the Special Ops. This is your acquired immunity. These are the master strategists, your B cells and T cells, and they get called in for specific threats. And here's the game changer. They learn, they adapt, and they remember enemies forever. This is the team that vaccines are designed to train.
Now, to fight an enemy, you first have to be able to identify them. In immunology, the way you do that is by spotting their uniform, and that uniform is called an antigen. It's basically any substance that your body looks at and says, hey, you're not from around here. That uniform is the signal that kicks the whole defense system into high gear.
Okay, so once that alarm goes off, the Special Ops team gets to work creating antibodies. And the absolute best way to think about these amazing little Y-shaped proteins is as custom-made handcuffs. Seriously, each antibody is built to latch onto one and only one specific antigen. They are precision-guided weapons that find their target, lock on, and tag the enemy for destruction.
But here's where it gets even cooler. Not all antibodies are the same. They're like different agents with specialized jobs. You've got IgM, the first responder. It's the biggest one, and it's always the first to show up at a new infection. Then there's IgG, the veteran. It's the most common and provides that all-important long-term memory. And this is so crucial because finding IgM tells a doctor you have a current infection, while finding IgG means you've either had it before or you've been vaccinated. Then you have IgA, the secret agent, doing undercover work in places like saliva and tears. And finally, there's IgE, the alarmist, the one responsible for those sometimes dramatic allergic reactions to things like pollen.
Okay, so we've got a handle on the battlefield inside our bodies. But how does that help a scientist in a lab? How do they peek inside and figure out exactly which war is being fought and who's winning? Well, this is where we leave the battlefield and step into the crime scene investigation.
You know, the entire field of serology, which is just the fancy word for studying this stuff in your blood, it all boils down to one super simple idea. A detective can prove who was at the scene of the crime by finding one of two things, either the bad guy himself or the specific tools used to catch him. For us, that means finding either the antigen, the enemy's uniform, or the antibodies, those custom handcuffs your body made just for them.
All right, let's start with a classic detective technique. It's simple, it's visual, and believe it or not, it's still incredibly powerful. It's called agglutination, but we're going to call it the clumping test.
So here's how it works. A lab tech takes a tiny drop of your blood serum. Now that serum might be full of your custom-made antibody handcuffs. They then add a drop of a liquid that has known enemy uniforms, specific antigens. They give it a little mix, and if your antibodies are in there, they're going to do what they were born to do, grab onto those antigens. When millions of handcuffs grab millions of uniforms, they all link together and form these visible clumps. It's a brilliantly simple idea. If you see clumps, it's a positive ID. It proves your body made the handcuffs because it saw that enemy before.
Now let's move on to a more modern, high-tech tool in the detective's arsenal. It's called an ELISA test, and I want you to think of it like a flare gun that lets you spot an invisible enemy in the dark.
This one is a bit more involved, but it's such a clever process. First, the detectives set a trap. They coat a special lab plate with bait, either the known enemy uniforms or the known handcuffs. Then they add your serum. If the target they're looking for is in your blood, it gets caught in the trap. Next, they add a special detector antibody that has a little enzyme attached to it. This detector latches onto whatever was caught, creating a kind of molecular sandwich. And then, for the final step, a chemical is added.
And this is the big reveal. That little enzyme on the detector antibody acts like a trigger. It reacts with the chemical and produces a visible color change. It is literally like firing a flare into the sky to signal you found something. No color change? Nothing's there. A bright, clear color? Boom, positive ID. The enemy or the evidence of the enemy is right there.
So this immune system is obviously incredibly powerful and really, really precise. But because it's so complex, things can sometimes go wrong. So what happens when this amazing army overreacts to something harmless? Or even worse, what if it mistakes its own side for the enemy?
Well, this is called hypersensitivity, and it comes in a few different flavors. Type 1 is the false alarm. These are your classic allergies, where your body just completely freaks out over something totally harmless, like pollen. Type 2 is a misfire, where antibodies start attacking the body's own cells, which is what happens in a mismatched blood transfusion. Type 3 is kind of a clogging problem, where those antibody-antigen clumps we just talked about get stuck in places like your kidneys, causing big problems. And Type 4 is the delayed reaction, a response that takes a couple of days to even show up.
And that brings us to maybe the most tragic mistake the immune system can make: autoimmunity. This is the ultimate case of friendly fire. For some reason, the system gets confused. It loses the ability to tell the difference between self and invader, and it turns all those powerful weapons inward, attacking its own healthy tissues and organs.
Okay, so let's wrap this up with a final, fascinating detective's paradox for you. We've just learned that finding antibodies is the key to a positive test result. So, is it possible for a test to come back negative, not because there are too few antibodies, but because there are too many?
And the answer, incredibly, is yes. It's a real thing called the Prozone phenomenon. It turns out that in those clumping tests, if you have an overwhelming flood of antibodies, they can actually get in each other's way. They end up coating every single enemy uniform so completely that they can't link up to form those big visible clumps. It creates a false negative, a total diagnostic blind spot. And the solution is as weird as the problem itself. You just dilute the sample to thin out the antibodies and run the test again. It is a perfect final reminder of just how complex, how strange, and how endlessly fascinating the secret war inside all of us really is.